Hi, Michelle Glass here. Welcome back to another episode in our Chapter 6 Lecture 6 series. And our topic for this video is glycolysis. When we last left off, we were talking about the significance of passing electrons from glucose to oxygen, and we were focusing in on that electron transport chain, which we will come back and talk about, but toward the very end of our lecture series. So that's toward the end of our chapter. We were talking about the electron transport chain and looking at the significance of that NAD plus and NADH molecule in terms of our oxidation and reduction reactions. Our job here is to begin by looking at how cells make ATP starting with the glucose molecule. And the very first step in that process is a reaction called glycolysis. And if you want to, you can think about this as glucose lysis, and you can sort of mispronounce it if you want and call it glycolysis to help you remember. We're basically here breaking apart a glucose molecule. Now we need to know a few facts about glycolysis, so let's just go ahead and get those out of the way. Glycolysis is described as an anaerobic reaction. Anaerobic means without oxygen. So that will be different from what we've talked about in our first presentation, where we referenced oxygen as our final electron acceptor. We're not going to see it involved in this particular step. This process, glycolysis, is going to occur in the cytosol. Remember, the cytosol is the fluid of the cell, and we can also talk about it as the ICF. And we see that all cells, whether they're prokaryotic or eukaryotic, have cytosol. And so that's going to make glycolysis a universal process. We don't need to have any specialized organelles in order to perform glycolysis. We just need to have what are called glycolytic enzymes involved in glycolysis. And so we will see glycolysis in both prokaryotic and eukaryotic cells. So you can see it all the way from domain archaea, bacteria, all the way up through domain eukarya. Now, glycolysis will produce some ATP, but it is going to be a very small amount in comparison to what we can see being produced at our mitochondria. So this is typically, especially in a eukaryotic cell, not the primary method for making ATP, but it is a source of ATP for the cell. And we will see that in glycolysis, we're doing what is described as substrate level phosphorylation, as opposed to a process that we'll see later on in the mitochondria. Okay, so here is our basic reaction. So with glycolysis, we are starting with glucose. Glucose molecular formula is C6H12O6. So essentially, we see that it is a 6-carbon molecule. So I've given you your 6 carbons as those dots. In glycolysis, you have lots of different steps with lots of enzymes being involved in each one of those steps. All of that is diagrammed for you in the artwork of your textbook. We do not have to memorize all of those different steps, so I'm summarizing it here. When the cell is breaking down glucose, ultimately we're going to get 2 pyruvate molecules being produced. Each pyruvate molecule is a 3-carbon molecule, so we're taking the glucose, which is 6 carbons, and we're splitting it up into 2 3-carbon molecules, so we're dividing it in half. When we do this, we're going to see some ATP being produced. We already mentioned that there will be a small amount produced here. That ATP that we produce here is just 2 ATPs. We are keeping track of how many ATPs are produced at each step, so that is something that you want to write down and remember. And also we will see as part of this process that our NAD plus molecule is reduced, meaning that it is going to gain electrons, it's going to gain hydrogen. So we will see also that we're producing here 2 NADH molecules, which are going to be carrying our hydrogen. So we're pulling hydrogen off of our glucose. So that means our glucose is being oxidized in this case. Looking down at the bottom, I've talked about these as being pyruvate molecules. You'll also see it referred to as pyruvic acid molecules. So just keep in mind that's talking about the same basic thing for us. Okay, again, our glucose is being oxidized. Our glucose is losing hydrogen or losing electrons. And our NADH is being reduced. Is it gaining hydrogen or gaining electrons? Remember, this process is happening in the cytosol, so it's a universal. It's not required to have any specialized organelles, just glycolytic enzymes. It is anaerobic, no oxygen is required here, and no oxygen gas. And it is producing some AT, but it's a pretty small amount in comparison to what we'll see in the future. Okay, so that's it for glycolysis.